right, so usually the ion situation inside and outside of the cell is it's pretty stereotyped for neurons. So usually these are the values that we usually see for extracellular and intracellular fluid outside and inside of a cell. So therefore, we have typical equilibrium potentials for each ion, because remember that these concentrations go into determining the equilibrium potentials for the ions. Okay, so potassium has an equilibrium potential of -90. Sodium typically has an equilibrium potential of +60, and so on. Okay. Just to make sure that we reinforce this concept, I'm going to have you pause and answer these questions here. What does it mean? Put it into words. What does it mean for the equilibrium potential of potassium to be at -90 millivolts? What does it mean for ENA, the sodium equilibrium potential, to be at +60 millivolts? So go ahead and hit pause and answer those questions. Okay, so hopefully you were able to express the concept in words. It is hard. I always have to practice this lecture many times to kind of get the explanation clear. But hopefully you were able to do it to some degree. Moving on, I want to point out something very important and interesting about that table that you saw on the previous slide, which is now on this slide. I want to point out these concentrations for potassium and sodium. They're pretty bizarre if you look at it. So typically outside of a cell, there's very little potassium, and inside of a cell, there's a lot of potassium. And typically for sodium, you see a lot outside of the cell and very little inside of the cell, okay? This didn't happen by accident. It is the result of hard work by the sodium-potassium ATPase. So to remind you, I'm pretty sure you must have heard of the sodium-potassium ATPase somewhere else. Just to remind you, the sodium-potassium ATPase pumps out 3 sodiums for every 2 potassiums that it pumps in. And we're working against these ions' concentration gradients, so that's why it's an ATPase. It requires an energy input from ATP in order to achieve these very large concentration gradients, okay? I want to mention that the sodium-potassium ATPase is no joke. It is it can use up to 75% of a neuron's energy expenditure. So up to 75% of a neuron's energy expenditure goes into this process of pumping sodium and potassium across the membrane so that we can achieve these concentration gradients. The reason we put so much effort into this is that these concentration gradients are being set up so that we can then use them to generate electrical signals in the neurons, as you will see in the next set of lectures. Okay, so let's take a moment now to review some of the concepts that you have been introduced to. I'm just going to read through all of these and then let you pause to go ahead and answer them on your own time. So first question is, what do these particular symbols mean? Second question, take a look at this figure. What is this cell's extracellular potential? And then this question right here, which is actually a 2-part question, how do you express each of these as an equation? So 1, the inside of the cell is 50 millivolts more positive than the outside of the cell, and 2, chloride is at equilibrium when the inside of the cell is 63 millivolts more negative than the outside of the cell. So go ahead and hit pause to answer those questions. Okay, so hopefully you were able to make it through those questions. We will be discussing them in class. So that wraps up our discussion of equilibrium potential. These are your learning goals. Basically understand what equilibrium potential is and how it's determined. And then also know the typical equilibrium potentials for those main ions that we see around the cell. Here's your vocabulary. See you in the next video.